Plant parasitic nematodes are recognized as one of the greatest threats to crops throughout the world. Nematodes alone or in combination with other soil microorganisms have been found to attack almost every part of the plant, including roots, stems, leaves, fruits and seeds. Plant parasitic nematodes are microscopic roundworms that must have living host tissue to feed upon in order to grow and reproduce. Every cultivated plant species has at least one nematode reported to parasitize it. In turn, most agricultural sites are infested with at least one species of plant parasitic nematode. The economic consequences of crop loss due to nematode-borne diseases are many and varied. These involve reduction in quality and quantity of crop yield. Cyst and root knot nematodes are the two most widespread. Economically important plant parasitic nematodes and cause serious loss on all crops. However, because most of them live in the soil, they represent one of the most difficult pest problems to identify, demonstrate and control. Their effects are commonly underestimated by farmers, agronomists and pest management consultants. But it has been estimated that some 10% of world crop production is lost as a result of plant nematode damage. Some of the most commonly practiced methods will be discussed, including crop rotation, the use of resistant and tolerant cultivars, cultural practices and chemicals. It is important to stress that the most appropriate control method will be determined by the nematode involved and the economic feasibility of implementing the possible controls. This will be briefed in the coming sessions. Key nematode of cereals and millets. Cereal constitute the world's most important source of food. Among them, rice, wheat, maize, sorghum occupy the most prominent position in terms of production and source of nutrition, particularly in developing countries. It has been estimated that about 70% of land cultivated for food crop is devoted to cereal crops. Rice. Rice is the most important crop in the world, predominantly in Asia, and accounts for up to 90% of the world's rice grown and consumed. Different systems of cultivating rice have been evolved to suit specific environments, namely irrigated, rain-fed, lowland, deep water, tidal wetlands and upland, and plant parasitic nematodes have adapted to each cultivation system, be it as a foliar parasite or root parasite. Now we will see the economic importance and its distribution. Depending on their parasitic habits, nematode pest on rice can be divided into two groups. The foliar parasites feeding on stems, leaves and panicle, and the root parasites. Well-known foliar parasites of rice include Aphalangoides baezici, Aphalangoides necales and Aphalangoides parent-necales. Aphalangoides baezici causes white tip disease in rice and is found widely distributed throughout the rice growing areas of the world. Here the disease is seed-borne and infected seed act as a primary source of infestation. The disease symptom caused by Aphalangoides baezici in rice are chlorosis of the leaf tip that gradually turns to necrosis. There is also distortion of the flax leaf that encloses the panicle. Other symptoms include stunted growth, reduced vigor and panicles bear small and distorted grains. The disease is now easily controlled by seed treatment and use of resistant cultivars, thereby inhibiting such high losses. Other major foliar nematodes pest in rice is Titulungus angustus, which is mostly limited to the South and Southeast Asia, where deep water and lowland cultivation systems are practiced. Titulungus angustus cause Uphra disease in rice, and the most prominent symptom of infected plants are chlorosis, twisted leaves, and swollen lower nodes. The base of the infected panicles is usually crinkled with empty shriveled glooms. The panicle head and flag leaf are twisted and distorted. Annual crop loss due to Uphra disease has been estimated at 4% in Bangladesh and 10 to 30% in Assam and West Bengal in India. However, soil density of Titulungus angustus has declined as area sown to deep water rice has reduced. Nematode root parasite of rice include migratory endoparasites, sedentary endoparasite, that means cyst and root knot nematodes, and ectoparasites. Hispanilla, well known as the rice root knot nematode, have been reported mainly from the rice growing region where lowland and deep water irrigation is practiced. As many as 7 species are reported to damage rice, but Hispanilla oryza being most commonly reported species. The symptoms of infection caused by this species Hispanilla are not specific and include poor growth, leaf chlorosis, reduction of tillering and yield. Nematode invade roots and penetrate through the cortical tissues, causing cell necrosis and cavities in infected roots, which in later stages turn brown and rot. The crop losses due to Hispanilla species have been estimated to about 25%. Several species of root knot nematodes have also been reported on rice, the key species being Melodogeny graminicola, which is mainly distributed in South and Southeast Asia, upland, lowland, deep water and irrigated rice is prone to damage by Melodogeny graminicola. Swollen and hooked root tips are the most prominent and characteristic symptom shown by Melodogeny graminicola and Melodogeny oryzae. Typical above ground symptoms include stunting and chlorosis leading to reduced tillers and yield. Crop loss at 2.6% for every 1000 nematodes have been estimated due to Melodogeny graminicola. Major methods of control. Most effective means of preventing yield loss due to Aphalangites species is by using clean seeds, and most effective ways for reducing crop loss include fumigating seed with methyl bromide, gamma radiation and seed dressing with effective nematicides, hot water or chemical seed treatment. Seed treatment is a very important step which is usually done with hot water. It requires free soaking seed in cold water for 18 to 24 hours, followed by immersion in hot water at 51 to 53 degrees Celsius for 15 minutes. Other control measures for reducing the crop losses due to Aphylangite species include using resistant or tolerant cultivars and low seed planting densities. Many different methods are used to control Uphra disease in rice caused by Titulungus angustus. These include removing the infested stubble or straw, using early maturing cultivars, burying crop residues. Management of Hispanilla species comprises various methods, namely soil solarization, fallowing, weed control, use of resistant cultivars and rotation with non-host plant. Flooding of soil reduces the population density of this nematode. Root knot nematodes in rice cultivation can be managed by adopting soil solarization and use of resistant cultivars. Wheat. Wheat is the third largest cereal staple with production of 633 million tons each year and the three largest producers of China, India and the USA. The primary nematodes of global economic importance on wheat are cereal cyst nematode, that is means Hydrodera, and root lesion nematode Pratyluncus. Economic importance and distribution of this nematode. The most economically important and globally recognized nematode on wheat is the cereal cyst nematode, that means CCN. The CCN complex comprises of a group of 12 valid and several undescribed species, but the most economically important species is Hydrodera avene. Hydrodera avene is the most widely distributed and damaging species on cereals cultivated in more or less temperate region and is cosmopolitan in nature. Two other parasites of the same species are Hydrodera philippivi and Hydrodera avene, which due to its widespread distribution also forms an economically important nematode on cereals. The above ground symptom produced by CCN are often confused with nitrogen deficiency or poor soil symptom. The foliar symptoms occur early in the season as pale green patches. These patches vary in size from 1 to 100 meters square, followed by yellowing of the lower leaves of the plant, which generally bear few tillers. The root damage caused by cereal cyst nematode exaggerate the effect of any other abiotic stress. For this, we can quote example as water or nutrient stress. Migratory endoparasites of the genus Pratyluncus are the second group of economically important nematodes on wheat production system. 8 species of Pratyluncus infect small grains and the 4 most important species Pratyluncus thorni, Pratyluncus neglectus, Pratyluncus penetrans, Pratyluncus crinatus or polyphacus and have a worldwide distribution. Pratyluncus thorni is the most extensively studied of this species on wheat and has been found in Syria, Yugoslavia, Mexico, Australia, Canada, Israel, Iran, Morocco, Tunisia, Turkey, Pakistan, India, Algeria, Italy and the USA. With respect to economic importance of Pratyluncus thorni causes maximum yield loss in wheat due to its wide distribution. On an average, about 30 to 70% of yield loss is incurred due to Pratyluncus thorni, Pratyluncus neglectus and Pratyluncus penetrans appear to be less widespread and damaging on cereals compared to Pratyluncus thorni. Major methods of control to this nematode. The major methods adopted in controlling both cereal cyst nematode and root lesions nematode are crop rotation with non-host crops and also genetic host resistance, since cereal cyst nematode is host specific. Rotation with non-cereal offers better chance to reduce nematode density. But Pratyluncus is largely polyphacous and hence crop rotation may not help much to bring down lesion nematode load in the soil. Using resistant cultivars have proved to be one of the most cost effective and straightforward methods for nematode control with respect to cereal cyst nematode and Pratylengus. Genetic resistance is favored with the addition of genetic tolerance. The identified source of resistance to Heterodera avene have been found predominantly in wild relatives of wheat in the Aegilops genus and have already been introgressed into hexaploid wheat backgrounds for breeding purpose. Unlike cereal cyst nematode, no commercially available sources of cereal resistance are available to Pratylengus thorni. Maize crops. Out of all the cereal staples, maize has the highest production. It is grown predominantly in tropical and subtropical region of North America, Asia and Europe from the three largest producers of maize. Out of more than 60 nematode found associated with maize, the ones that pose important limiting factor in maize production from all over the world include the root knot nematode caused by Millets species, the root lesion nematode Pratylengus species and the cyst nematode Heterodera species. Most of these have been recorded from roots or soil around maize roots with information on the biology or pathogenicity of many of these species not readily available. Now, we will see the economic importance and distribution of this nematode. Root knot nematode, Millets species, is the most economically important plant parasitic nematode species causing maximum loss. After root knot nematode, Pratylengus species along with Millets and Hoplolaemus were the most frequently reported genera on maize. Several species of root knot nematodes including Millets incognita and Millets javanica have been detected and destroying levels in almost all maize growing region of the world. Millets africana and Millets arenaria have been reported on maize in India and in Pakistan. Foliar symptoms include reduced growth of plants, yellowing of leaves and patchy growth. In contrary to root knot symptoms observed in other host plants, in maize root galls may be small or large, terminal and subterminal or in certain cases totally absent. Hence, maize has often mistakenly been considered a poor host or even a non-host for root knot nematodes. Though the opposite is true and soil sample is such maize grown region often exhibit more than threshold levels of nematode parasites and bring about yield loss. Lesion nematodes have a cosmopolitan occurrence in maize fields. Several of these species have been reported to be associated with poor growth and yield reduction. A study conducted on nematode pest of maize also reported direct relationship between Pratylengus hechinensis and Pratylengus scribneri and maize yield loss. Nigeria and Pratylengus brechuris has been reported to be responsible for a 28.5% yield reduction. Other indirect evidences suggest that lesion nematodes are important limiting factors in maize cultivation. The third group of nematodes of importance for maize are the cyst nematode. Although more than 9 species of cyst nematodes have been found associated with maize in subtropical and tropical countries, only 3 species, namely Heteroderaceae, Heterodera avene and Punctodera chalcosensis, are considered economically important. The above ground symptoms are relatively non-specific. Heterodera GA infested plants exhibit reduced growth and are stunted and pearl green in color. On the other hand, Punctodera chalcosensis is limited in distribution to Mexico and shows almost same symptom as those for Heterodera GA. It is being estimated that maize growth and yield are suppressed by 13 to 73% in the presence of Heterodera gea. There is little information available about the importance of Heterodera avenae on maize, but this could be very important in wheat and maize. Functodera chalconensis is highly restricted with only two plants, Gea maize and Gea mexicana, and therefore damage by Functodera chalconensis can be severe and is dependent on cultivar susceptibility, nematode density and adequate soil moisture level. Major methods of control of this nematode. Cultural practices such as crop rotation, planting time, application of organic amendments, biological control have been proved to be effective and in reducing nematode population in many cases. But these practices are genera and species specific. For instance, in Mexico, early sowing dates and adequate fertilization reduces damage caused to maize by Functodera chalconensis. Crop rotation may seem to be a promising option as maize has been suggested as a good rotation crop that can help reduce population of some nematodes. But little is actually known about the effects of rotation on root knot population densities in a maize crop. Ineffective crop choices in crop rotation or sequences where maize was involved demonstrate the dangers of susceptibility of maize to various species of nematodes. Although several biological control agents have been investigated against many species of plant parasitic nematodes of importance to maize, none of these biocontrol agents can be used economically at the present time in extensive cereal crops. Sorghum. Sorghum is subjected to damage due to various species of nematodes. This includes sting, Belonomyia longicordatus, stuffy root, Paratrichoderus species, Lesion pratylengus species and ring Cricolonymla species nematodes. Unless the crop has been monocultured for several years, damage from these nematodes to sorghum is usually not severe. The potential for nematode problem in sorghum could be due to cropping history of the field. For example, repetitive cultivation of sorghum or other grass crops equally nematode susceptible crops for 2 or more years. Above ground symptoms of nematode injury include reduced growth, thin stands, premature wilting and nutrient deficiency symptom and chlorosis. Above ground symptoms of nematode damage vary greatly in shape, size and distribution. The ground symptoms such as small knots on roots or stunted and swollen root tips can also be observed. Management practices to control this nematode. Crop rotation. Sorghum is often used as non-host plant in crop rotation system rather than being used as the object of a nematode management program. Crop rotation is the most effective means to manage nematodes in sorghum. Many nematodes have been found in association with small grain cereals, out of which only few are considered economically important. Those of importance include cereal cyst nematode Heterodera species, root lesion nematode pratylengus species, root knot nematode Melioidogenous species, seed gall nematode Anchinotriticea and stem nematode Tytelungus dipsaceae, which have wide host range including rice, wheat, barley, maize, etc. Management of nematode pest of cereal crop is important in order to ensure food security and to feed the increasing human population. With this in mind, It is essential that the full spectrum of crop production limitation is considered appropriately, including the often overlooked nematode constraints. A proper approach in this regard would be to use integrated pest management system involving combination of methods, or may involve only one of these methods. There is a need to have an insight into the economically important nematodes on small grains, their distribution, damage potential, and the management option that exist for their control.